Now starting Chapter 10. The title of Chapter 10 is DNA, Structure, and Replication. Now this is our very first slide of Chapter 10 of any significance. Now at the very bottom you're going to find three bullets. These three bullets deal with the function or the importance of DNA. Number one, stores genetic information. So that's what the DNA actually does. It stores genetic information. Very much like a hard drive stores information, the DNA stores genetic information. Second, copy. You're able to actually send information as a copy from one to another to another to another. So we're able to copy the DNA. The last one, we are allowed to pass it from generation to generation. So when your kids pick up your genetic information, they've actually picked it up from your DNA. So you're able to pass on your genetic information from generation to generation. Okay, next slide for chapter 10. Now, we already know this, so this is a little bit of a repeat from previous chapters. We already know the most commonly missed question will be, what is a nucleotide? And if you recall, a nucleotide had three main parts. So this is from a previous chapter. Nucleotides have a nitrogenous base, they have a phosphate group, and they have a pentose sugar. So our previous knowledge is coming back here. So nucleotides have a phosphate group, a nitrogenous base, and a pentose sugar. Now, at the very bottom, we already know this as well. We know that for DNA, our nucleotides, our nitrogenous bases, are A, T, G, and C, mentioned at the bottom. Next slide. We already know that we have a phosphate sugar. So we already know this as well. Next slide. Now, this is a video, which I do not care for, but it is something that we've already mentioned before as well in another chapter. We need to know that DNA is double-stranded, shown by the left picture, whereas RNA is single-stranded, shown by the right picture. So that's the take home so far. The other thing that we learned before is that DNA contains thymine, whereas RNA contains uracil. So again, a prior knowledge coming back. Next slide. Okay, don't care. Now, this is a picture from a previous chapter that's now put back in this chapter. Here we see our nucleotide, the phosphate group, the pentose sugar, and in this example they're showing thymine as your nitrogenous base. So this is prior knowledge coming back. Now what's new this time is what's coming up shortly. Okay, don't care. Don't care anymore. Don't care. Again, we've already had this in the previous chapter. Remember I told you this was the most commonly missed question. What are the three parts of a nucleotide? The nitrogenous base, the pentose sugar, and the phosphate group. Next slide. Don't care. We already know this too. We know that the sugar for DNA is deoxyribose. So that's the sugar for DNA. What do you think the sugar for RNA was in the past? Ribose. So that's the difference. Okay. We already know this too, that thymine and cytosine are single rings, mentioned previously, and A and G are double ring structures. So this is a repeat from the past. One more time. Thymine and cytosine are single ring structures, and adenine and guanine are double ring structures from the past. Okay. We already know this slide as well from the previous chapter. We know that RNA has uracil, whereas DNA would have thymine. So again, a repeat. Okay, don't care. Don't care. Now this slide we're going to stop for a moment on is because the two gentlemen on the left, they won the Nobel Prize in terms of figuring out the structure of DNA. The lady on the left, Rosalind Franklin, well, she didn't get the Nobel Prize. All she really discovered is through X-ray diffraction, she discovered that purines and pyrimidines go together. So something with a 2 ring goes with a 1 ring. So that way we know that adenine must pair with thymine, guanine must pair with cytosine. That's what Rosalind Franklin figured out. Next slide. Okay, don't care, don't care, don't care. Now this slide I do care, so let me give you a page number. We're now on page 174. So page 174. Now on this page, the picture I care for really is the middle picture. If you look carefully, you're going to find on one of them that all the oxygens are facing upward. So if you look at the pentose blue sugar there, the pentose blue sugar, the blue sugar shows oxygen on the left going up, and then the oxygen on the right is going down. The reason for that is because DNA is considered anti-parallel. Now what does that mean? It means that one side of the DNA molecule goes in one direction, whereas the opposite side goes in the opposite direction. So we have got one side going up and one side going down, and that's called anti-parallel. The other thing previously mentioned, and now we're really stressing it in this chapter, is the number of hydrogen bonds between the sugars or the bases. Well, for A and T, we see 2 hydrogen bonds. So between A and T, 2 hydrogen bonds. How many bonds between G and C? 3. So 3 hydrogen bonds between G and C, 2 between A and T. Okay, next slide. That was page 174. Here is an enlarged view of it. Okay. All right, still skipping. We'll come back to this video. It is worthwhile looking at, but it's worthwhile looking at after I get to a certain point. Next slide. Let me back up for a second. Now this video, which I can't play while I'm making a video, but this video is of value to you because what it does is it tells you how DNA replication occurs. So how DNA replication occurs. The DNA strand, as you see here as a double strand, will open up, and one strand will serve as a template and one serves as a lagging strand, and we'll see that later. Okay. Here's another picture that we're going to come back to as we watch our video. This is now page 175. So page 175. Now this is sort of part of that video that we skipped a moment ago, but if you look carefully here, we see the double strand at the top opening up, and then we have 2 new ones. In this case, I guess they're gold in color. The new ones open up as well here. So what we have here is an old strand of DNA opening up. Half of it is new, half of it is old. So a brand new DNA will open up and make 2 new DNAs, and the 2 new DNAs will have an old strand acting as a template to make a new strand. So there's going to be an old and a new within the new DNA. Now that has a name that's called semi-conservative. So when I have a new DNA having half old and half new, that's called semi-conservative. Page 175. Okay, I'll come back to this when we see our other pictures. So we'll come back to this. Don't care. Again, videos I do want you to watch, so make sure you're watching the videos that go with Chapter 10's PowerPoints. So make sure you watch the videos, but I'll go over them in a little while. Here's another video that is worthwhile watching. Now, the magic of this particular slide and this particular video that we're going to see is that one strand is called the leading strand. Because when you open up the DNA, and we already said that the DNA is double-stranded, it is also directional. All right, so one strand is going to be leading strand, the other one is the lagging strand. Now, one of the chemicals that I passed up intentionally because we're not watching the videos yet is this guy right here called DNA polymerase. It's an enzyme. So DNA polymerase. So now DNA polymerase can only add in the direction of 5 to 3. So it can only add pieces in the direction of 5 to 3. Now what that means is that the leading strand is easy to make another copy of because it's already open at one end. It's like a zipper. It's easy to start at the end where it's already an open end. So DNA polymerase will add pieces to make the other strand from the direction of 5 to 3. Now the top piece that you see here, called the lagging strand, it's got a problem because the lagging strand doesn't have an open end yet. It's way somewhere still coiled on the far right of this picture. So there's already a coiled part that's not open or accessible yet. So that means that for the lagging strand, I won't be able to make it in the same way. So this is going to require some small pieces. And in the picture there, which is the first freeze frame of the video, you see the little red pieces. Those red pieces are called RNA primer. So those red pieces you see in the picture right now, those are called RNA primer. It actually needs a starting piece. So RNA primer. The other thing about the lagging strand, the one at the top, is it can't be made as one uniform piece. Because it has the RNA primer in certain red places there, the pieces are made in fragments. So the lagging strand has pieces called fragments that have a name, and it's called Okazaki fragments. So one more time, the lagging strand made in pieces, starting with the RNA primer in red, those little pieces are called Okazaki fragments. It is mentioned in the video, within this video. All right, next slide. Don't care. Don't care. Okay, don't really care. Okay, don't really care about these two items. Now, this concept of transcription and translation, that is different and aside from the DNA replication. So before I go any further, it is very important that you first get down DNA replication, because that's one whole story of itself, and then you can move on to this slide, which is transcription and translation. So it won't make sense until you first get DNA replication down as a separate story, and then we move to this slide, which is transcription translation. Okay, moving forward. Notice there's actually a video for that part as well. So transcription and translation fall under a section called protein synthesis. Now, before I go any further, again, this is another video that we need to be able to watch. Now, the catch about this one is when it says protein synthesis, we're going back in time to something from your flashcard. Protein synthesis occurs on the ribosome. So transcription and translation will eventually deal with ribosomes. Okay, moving on. Next slide. Now, again, don't confuse RNA or DNA replication story. That's one thing. And now we're looking at transcription, translation being a separate thing. Now, you do need to know this picture, so let me give you a page number. It's page 176. Now, why you need to know this picture on page 176 is because you need to know where you are and then what's going on. So from this picture, we see transcription occurs in the nucleus. So that's why it's a picture story. Page 176, transcription occurs in the nucleus. What goes on? Well, in the nucleus with transcription, we're taking DNA and turning it into RNA. So DNA being turned into RNA, that's transcription taking place in the nucleus. Now the bottom half. The bottom half says translation. Now where is translation going to occur? It's going to occur in the cytoplasm. Now what's happening here? Well, the RNA has left the nucleus. It's going to go to the ribosome. So RNA will go to the ribosome. And in this cytoplasm, the ribosome is going to convert it into a protein chain. And remember, amino acids make up proteins. And how many different kinds of amino acids are there from the past? 20. So that old news, that old notes come back here. So translation occurs in the cytoplasm, and it's taking the RNA and turning it into a protein. Okay, next slide. Okay, now this is now page 177. So page 177. Now from the top here, we see different genes. We see a green one, a yellow one, and a blue one. So these are different pieces of genes. And you already know the definition of a gene. A gene is a sequence of DNA that codes for a specific protein. So a sequence of DNA that codes for a specific protein. What this picture is saying, that by way of transcription, by way of translation, we're going to take whatever genes we have, and we already know that genes have our traits on there, whether they be height as a characteristic, blue eyes, black eyes, whatever we have. So we have our characteristics, we have our traits, all embedded in the gene, and somehow that translates out. So we have transcription, translation, eventually making a protein. Now if we look at our picture here, we see a DNA strand. In this example, they're picking one of the sides. So of the two strands, they're picking one of them as a template. And in this example, the template reads AAA for DNA, and then the corresponding RNA would be UUU. The next set reads CCG, and the corresponding for the RNA in red would be GGC. And each one of these little three letters there on the RNA, each of those three letters on the RNA, in this case would be UUU, those are called codons. So a set of three, and we call them nucleotides, so the set of three nucleotides on an RNA is called a codon. Okay? So a set of three nucleotides on RNA is called a codon. Now the codon gets translated, just like you translate a language, it gets translated to a specific amino acid. So in this example here, the DNA strand of AAA converts over to UUU for RNA, which then converts over or translates over into a amino acid. In this case, it's a square. Okay. So if we look at the neighboring one, the DNA was CCG. The RNA that it relates with is GGC, and then that translates over to a circle in terms of its amino acid. Remember, there are 20 known different kinds of amino acids, all accounted for this way. All right, next slide. Actually, let's go back one more before we go further. What happens if I change one of these letters? By changing a letter, so let's say I have AAA, and then of course it translates to UUU. What if I change the first letter? Instead of an A, it's something else. Well, here's the rule to keep in mind. Changing the first two letters is very dramatic. It can cause big changes. Let me give you an example. The difference between a red blood cell of a human and the red blood cell of a chimp is only one letter difference. So all of our DNA in a chimp that deals with a red blood cell, all the DNA in us as a human that deals with a red blood cell, the only difference is one letter. That's how dramatic it can be in terms of making a red blood cell for a human versus a red blood cell of a chimp. So the first two letters are very, very dire. You change the first two letters, you can have very big consequences. Now changing the third letter of that codon, so in this case the last U, by changing the third letter, we have some reprieve. That last letter of the codon is called a wobble. That is correct. W-O-B-B-L-E. Wobble. It turns out to be a technical term. So wobble is the last letter of that codon. Now the thing is, nature has set some allowances. So just like you have pass-fail basis sometimes in some classes where all you got to do is pass and that's it. Well, there's an allowance there. It says anything above a 70 is a pass, that kind of thing, right? Well, in the same way here, nature says you don't have to be 100% perfect. You can have the first one right, first letter correct, second letter correct. Maybe you change the third letter a tad bit. We have some allowance, and guess what? That wobble is the allowance. Where if you don't have a U in the last letter, let's say you put an A in there instead, you'll still end up getting the same thing. So in some cases, that last letter is a wobble. By changing the last letter, you don't necessarily change the kind of amino acid that will come out. So nature has a built-in allowance there, a little wobble. So the last letter is sort of relatively protected, but changing the first two letters, you're typically screwed one way or the other. Okay, so it's very dramatic effects on the first two letters, not so in the third. Next slide. Okay, I already given you the definition of a codon. It's the set of 3 nucleotides on an RNA. Moving on. Okay, I don't care for this slide. Now, we do have a chart available on page 178. Now, you do not have to memorize this chart, but if a chart is given to you, you should know at least how to use it. Now, what do you have to memorize off this chart? You need to know the 3 stop codons that you see at the top. So you do have to memorize those 3. So the 3 stop codons are UAA, UAG, UGA. So those are the 3 stop codons you have to memorize. UAA, UAG, UGA. You also have to memorize the green one, which is the start codon. The start codon is AUG. So in order to start the sequence off, you've got to have a codon that starts with AUG. Now the AUG is not only a start, but it's also for the first kind of amino acid that's called MET. Now the full name is called methionine, which I don't care for. You can easily abbreviate it as MET, and that's fine with me. So AUG is either a MET or it is a AUG as a start codon. So those are the ones you have to memorize. Now let's go look at this chart again on page 178 for another point. Let's look at the very first corner box. The first corner box you see UUU, and then right below that you see UUC. Notice I only changed the last letter. Okay, so UUU and UUC. I only changed the last letter, and yet I still make what? I still make phenylalanine, which is PHE. Still. Okay, let's take the box just below that. We have CUU, CUC, CUA, and CUG. Notice I'm only changing the last letter, either CUU, CUC, CUA, or CUG. But still, no matter how I change the last letter, it still makes leucine, which is the amino acid. Okay, does that now make sense to you with my previous example? By changing the last letter, it doesn't necessarily change the amino acid. If you go across any of them, such as we had earlier UUU, and now if I move over to the right of that, I now have UCU for serine, and look what happened. I didn't change the first letter, but I did change the second letter from a U to a C, and now it's UCU, which makes serine. So changing the first letter or changing the second letter can be very, very dramatic. All right, next slide. Don't care. Don't care. Now, this is page, let's see if I have a picture of that. Yes, I do. Page 178 again. And notice it says I've got a pig here with a yellow snout. Well, they actually introduced a gene into this pig that makes his nose not pink, but makes his nose yellow. Okay, glow like a firefly yellow. Now, why is it important? Because this whole thing called the genetic code, which I don't care for you to memorize at all, well, what this says is that this chart that we looked at a moment ago, it's true across the entire animal kingdom, plant kingdom, any kingdom. So all of life on the planet Earth uses this form of genetic code. So whether it's a plant, an animal, or whatever, it uses this, even bacteria uses this genetic code. So this genetic code is universal. And that's what they're getting at. So you can actually take a firefly gene, put it into a pig, and you get this outcome. Okay, moving on. Okay, now we're looking at the next possible steps. So we've gone through the overview, and now I'm on page 179 talking about transcription. Now, from this slide that I'm on now, you'll see that we have something brand new. It's now called RNA polymerase. That's because, as I said before, this starts off a whole new process. Do not confuse what we're doing now with the DNA replication with the other original videos that we saw before. So don't confuse the two. Now, transcription, we saw earlier, is found in the nucleus, and we're taking DNA and converting it over into RNA. In order to do that, we need an enzyme called RNA polymerase. So converting DNA to RNA requires RNA polymerase. Now the only picture I really care for is page 179, and it looks like this at the bottom. So here we have a double-stranded DNA in blue opening up and allowing us to make the red RNA. And as you can see, anywhere there is supposed to be a T, it is now being substituted with a U. So RNA, of course, has uracil. So wherever there's supposed to be a T, it gets substituted with a U. Okay, that's page 179. Now, Nimish, what about the stuff that's on the far right? Well, all I care there is just know the 3 stages. I don't care for the rest of the picture. So initiation, elongation, termination. Just memorize those 3 steps. That's all I care for. So initiation, elongation, termination. I don't even care for the picture for that part. Next slide. Here's a blow up of page 179. Again, you can clearly see the DNA being converted by RNA polymerase into RNA. Don't care. Don't care. Don't really care. Don't really care. Don't care. Don't care. I do care, but we're going to do it by way of a picture. I do care, but we'll do it by way of a picture, which is coming up. And that is this page. Page 180. Now on page 180, we see this picture showing you a blue DNA at the top. Now, DNA is 95, or actually 98, let me correct that, 98% garbage. And what I mean by 98% garbage, by the way, that would be very important to memorize, that it's 98% garbage. Now, what they mean by that is non-coding. It doesn't code for anything. So 98% is non-coding. It's just evolutionary genetic information. So what we need to do is get rid of some of the stuff. Just keep the fine points. So what we do here is we get rid of the introns. So as you can see in the picture here, introns, which are in the light blue, the introns are removed as garbage, and the exons, which are dark blue, are spliced back together. So we remove the introns, we keep the exons, and the exons are spliced back together, are joined back together. Now, the textbooks often call this RNA splicing. Okay, so RNA splicing is another name for this picture. Okay, so RNA splicing, we remove the introns as trash, We take the exons and splice them back together. Again, notice it's occurring where? It's occurring in the nucleus, and it's part of transcription. Next slide. Okay, now we just finished transcription. We're now looking at translation. Now, translation requires few ingredients. So on the picture of page 181, we're going to look at the ingredients that I need in translation. Okay, so I'm moving forward up to page 181. Now on page 181, I see this picture of a tRNA. Now the tRNA has two main parts. The top is where the amino acid will attach. So that little blue spike that you see, that is where the specific amino acid will attach. So a specific amino acid at the top. The bottom correlates to anticodon. So what do you think anticodon will attach to? The anticodon will attach to the codon of the RNA. So we'll see that on the next picture. So the anticodon will attach to the codon. Let's go look at that next. Okay, moving forward. On page 181 at the bottom, first I see the ribosome. And remember, we have two types of ribosomes mentioned long back. We have the free ribosome, and we have the bound ribosome on rough ER. Yes, that's all old news coming back. Now, a ribosome's got two parts, the large part and the small part. Some people care for that, so I'm just letting you know. So the ribosome's got a large part and a small part. Now, the ribosome has two towers, as you can see in the picture. The P tower, or the P site, that stands for promoter. So the P site stands for promoter. The A site stands for add-on, and you'll see that in a picture in a moment. So the P site is a promoter, and the A site is add-on. That's where new things come in. Now let's look at a picture on the far right here. Here we have our mRNA, that red ribbon there. That red ribbon is mRNA. Now the M stands for messenger RNA. So the mRNA. And yes, it's going to have our codons. So like AUG that you saw earlier, it'll have some of those stop codons also. So there are different codons along that entire red ribbon, that mRNA. Then we have our ribosome, which is our little blue thing. And then we have our tRNA bringing specific amino acids. Now in this picture, I see 4 of them attached to tower P. So you see 4 of them attached to tower P. Then you see 1 that's attached to tower A. Let's call this what, a square. So if you want to use it as a square, we see a square one attached to tower A. Now, what do you think is going to happen between tower P and tower A? Well, that single one on tower A is going to join with the ones that are on tower P, as in Paul. Now, how do I join amino acids together from the past? We've learned this already. Remember dehydration synthesis? That's right, old news coming back. So dehydration synthesis is going to join those two amino acids together. In this case, it'll be the circle one and the square one. And then all of that will now be moved over. So all of that will now move over. And we'll see a picture of that in a second. So everything that is on tower P will move over and join the square that is on tower A. So all 4 of those, the circle one, the triangle one, the square one, and the hexagon one, all of those will move over to that single square in tower A. Now here's what happens next. Okay, moving on. Here too we have 3 stages: initiation, elongation, termination. I really don't care for those. Don't care, don't care, don't care, don't care. Now this picture on page 182, I do care. We're going to look at this step by step. So here I have met. Okay, so met. And notice, what do I see in the red? The red is your codon. And remember, the start is called AUG. It's in red because it's a codon. What's the anticodon that goes with that? UAC. Then what happens? It moves over, since it's the only one, it moves over to tower P. Now, if tower P is now occupied, look what happened to tower A. It's open. It's open to go get another tRNA with a very specific amino acid at the top. And what decides which amino acid is going to be at the top? What shape it's going to be in this example from the book? That's going to be determined by the codon that you see at the bottom. So we have another 3 right there. We have sort of a U-shaped, a V-shape, another weird shape there. That's going to decide what specific tRNA and what specific amino acid comes in. So let's look at that next slide. So in this picture, it's multiple steps. We already saw the first step, which was step 1 in this picture, where a specific second one comes in tower A. Then, as I showed you earlier, or mentioned earlier, is everything on the P tower moves over to the A tower. Now, that arch red arrow that you see, that arch-shaped red arrow, that's letting you know that in step 2 of this picture, the dehydration synthesis has occurred. And everything in tower P, as in Paul, is moving over to tower A for the add-on. Now, what happens to the original item that was in tower P? Well, that pops off, as you see in picture 3 there. So the original P site, that original P site tRNA, goes off on its merry way. Everything is now on tower A. And if you do see a U-shaped arrow there now in red, that U-shaped arrow in red for step number 3 moves everything over. So all of that moves over to tower P. Tower A is now open, just like in step number 1, and this cycle will keep happening. And this cycle will keep going on, until it reaches what kind of a codon? Until it reaches a stop codon. And if you recall, there are 3 stop codons. All right, so there are 3 stop codons. Now let's move to the next picture, page 183. And the picture I care for is the one that's now on the slide. As you can see, at the very, very end of page 183's picture, step number 6 there, look what came in. A stop codon came in. Now, a stop codon does not bring a new tRNA or a new amino acid. Instead, a stop codon brings in what's called a releasing factor. So some tests and some books call it releasing factor. What do you think it really is? Water. So when the stop codon comes into play, you don't add any new items, and what you do is you bring in water as your releasing factor, and it cuts off, and then you have this blue chain of amino acids that you see in the picture. That is how polypeptide chains or proteins are actually made. Okay? So it's the DNA that decides how the RNA is going to look, and then the RNA will help decide how the protein is going to look. So if you mess up the DNA, you're going to mess up the RNA, which means you're going to mess up the protein as well. Okay, thus far we've reached page 183. Okay, next slide. Okay, now we're on page 184 dealing with mutations. Now, a mutation is a permanent change in a gene. That definition is a value. So a mutation is a permanent change in a gene. So mutation, a permanent change in a gene. Now, on page 184, they've provided an example of a mutation. So on this page we see CTT being the normal DNA, CTT. And that translates to GAA for the RNA, which then translates to a proper red blood cell with an amino acid called glucine, which is GLU at the bottom. But then on the right-hand side of this slide, we see the CTT now changed to CAT. Now notice which letter got changed? The second letter. So it's now reading what for the RNA? GUA. As a result, because I changed the second letter or the first letter kind of thing, it's going to change the amino acid dramatically. And now instead of glucine being the normal amino acid, we now have valine, VAL, valine, and now this person's got sickle cell anemia. So one letter change can cause a big change in the final outcome, especially if it's the second or first part of the codon. In this case, it was the second letter of the codon. Okay, that's page 184. So this is one type of mutation, and it's a permanent change in a gene. Okay, now let me back up for a second, and this is the slide you want to make a note of. The slide says types of mutations. We basically have two categories. We either have a substitution, which is what you saw a moment ago, where we changed one letter for another. So instead of having an A, it changed over to a U. All right, so that was one letter substitution. The other type that we're going to see in a moment is on page 184 called deletion or insertion. Now these tend to be more dramatic, and you'll see that on the next slide. They tend to be more dramatic. Next slide. Here's the example. At the very top, picture A, we see our normal sequence causing our normal sequence of methionine, lysine, phenylalanine, glycine, and alanine. That's your normal sequence. The second row for the base substitution, notice I have pretty much the same sequence, except instead of glycine, I have now serine for picture A. Why do I have serine? Because instead of a G, I now have an A for the first letter. And again, like I told you, the first letter and the second letter can be very dramatic if changed. So again, it's just a base substitution from a G to an A. So instead of glycine, it's serine. Now let's look at the bottom two. Let's look at letter B at the bottom. Letter B at the bottom says nucleotide deletion. Here I'm removing a letter. If I remove a letter, notice what happens. Instead of having 5 normal, remember at the top example, we see 5 normal amino acids? Well, now we only have 2 normal, and then we have 2 totally different. And we don't even have a 5th one at all now. So a nucleotide deletion actually is a bad thing. It now means we only have 2 normal ones and 2 bad ones and 1 totally missing. So we don't even have 5 anymore. So nucleotide deletion can be pretty bad. Then picture C at the very bottom. Picture C says nucleotide insertion. Here we're adding a letter. Again, notice adding one is just as bad. We do have 5, so we still have 5 amino acids, but only 2 of them are normal, and 3 of them this time are totally different than what we're supposed to have. So a deletion and an insertion can be very, very dramatic. Now that has a name. So when you add or subtract, it changes what's called a frame shift. So adding or subtracting will cause a frame shift mutation. And those are almost always very, very bad. Okay, next slide. That was page 184. Okay, I'm now on page 185 with this term, mutagen. Now, this word is often used wrong ways. A mutagen is simply anything that causes a mutation. So you can have mutations, a change, and that's caused by a mutagen. So a mutagen can cause mutations. But if that mutagen can also cause a cancer, then now it's called a carcinogen. And that's mentioned on this slide. So again, let's see the clarity again. A mutagen simply changes the DNA, not necessarily for good or for bad. But if it's for bad, the mutagen is now called a carcinogen. Okay. All right, next slide. Now, I'm on the second bullet of this slide. Okay, so I'm on the second bullet of this slide. The second bullet reads, mutations are one source of a rich diversity of genes in the living world, a diversity that makes evolution by natural selection possible. So this slide is letting you know that mutations are not always a bad thing. Matter of fact, mutations provide for genetic diversity. So just as we saw in a previous chapter, we learned that when it comes to the crossing over in meiosis 1 and crossing over allowed for genetic diversity in meiosis 1, same way here. Mutations allow for genetic diversity. The first bullet says, although mutations are often harmful, they can be beneficial both in nature and in the lab. That's how we actually make new creatures, new bacteria that we make and do things with. So we now have bacteria that eat oil. Well, nature didn't come up with the bacteria that eats oil. We did. So we took bacteria that didn't have the ability to eat oil and break down oil for us, and now we added genes to it, to the bacteria, and now that bacteria is able to eat oil and break down oil for us. So that's how mutagens work. They actually allow mutations to take place, which adds diversity to the world. Okay, next slide. Okay, now this one is a slide about viruses. Just be aware of the last two bullets about a virus. A virus is not considered to be alive because it doesn't have its own cellular mechanisms and it cannot reproduce on its own. So our viruses story that are going on right now with the coronavirus of 2020, well, that coronavirus of 2020 was created because it can't reproduce on its own. It uses a host, in this case animals and us, and it also does not have its own mechanisms. So that's page 186 with this slide. So not living, does not have its own cellular mechanisms, and cannot reproduce on its own. It needs a host. Okay, don't care. Here's what a virus typically looks like. Okay, again, I don't care too much for that. Don't care. Now this slide I do care. Viruses do attack bacteria. So yes, bacteria have viruses. Now they're called bacteriophages. So viruses that attack bacteria are called bacteriophages. And a picture of one on page 186, and that's what I'm moving to. Okay. So here's a picture on page 186 showing you a bacteriophage, which is actually attacking a bacteria at the bottom of the picture. And what's it inserting? It's inserting its own DNA. So the virus is putting its own DNA into that bacteria. Okay, that's page 186. Now, what happens, shown by this picture on page 187, is that the bacteria divides, and as it does, it actually makes not only more of itself as a bacteria, but it also makes more viruses. And eventually, the virus will be popped and released from the cell. It's going to kill the bacteria, but release more viruses. And that's what step number 4 is showing. So it has a stage where it actually makes more and more viruses until it pops the bacteria and releases the virus. On the right-hand side, you see that it's allowing it to make more bacteria. But it's making more bacteria containing what? containing more of the machinery and the genetic code of the virus. Page 187. And here it is, a picture of one showing you the viruses being released, and eventually it's going to kill this bacteria. Page 187. Okay, the next thing you need to know for this chapter is page 188. You need to know that plants have a virus, and the only example I care for is this one here called tobacco mosaic virus. So page 188, the only virus you need to know for the plant is tobacco mosaic virus. Okay. Don't care, don't care, don't care. Okay, I need to go back. This slide is based on page 190, and this one you do want to copy for the purpose of the test. So this one is a testable material. The first paragraph mentions all the RNA viruses. So many viruses like the flu, the common cold, measles, mumps, AIDS, polio, they are RNA viruses. Then the second paragraph mentions the DNA viruses like hepatitis, chickenpox, and herpes. Those are DNA viruses. So this is page 190, and this slide is of value. Okay, I'm skipping these slides, not important. Okay, don't care. Don't care. Don't care. Don't care. Okay, this one is of value. Notice that in winter months, more people tend to die from the flu, whereas in the summer, less people tend to die from the flu because it's a seasonal issue. Okay, next slide. Don't care. Okay, this one I do care. This one's based on page 191. And on this slide, we see AIDS, acquired immunodeficiency syndrome. It's caused by a virus called HIV, and it is a RNA virus. And it's often called a retrovirus because it actually takes the RNA from the DNA. So it's a little weird, and here's how it operates. The RNA goes into the host, puts into the DNA of the host, then it brings it back out as RNA again. That's why it's called a retrovirus. Okay, next slide. Don't care. Don't care. Don't care. Don't care. Don't care. Don't really care. Don't care. Now this picture that's coming up, I do care. It's on page 191. It is related to this slide that I currently have on the screen. There is a drug that's helping HIV, and that drug is called AZT. So someone might ask about that. So the drug is called AZT for HIV. Here's the picture version of AZT on page 191. You don't need to know the chemical formula of it. Okay, moving on. Okay, don't care. Now, this slide I do care because we previously have done it, and that's prions. If you recall in the past, prions were a protein-based germ that causes mad cow disease. So page 192, prions are proteins that actually cause mad cow disease. And we saw that earlier, page 192. And here's the picture that goes with that. So prions cause a number of brain diseases, and it causes mad cow disease. And in humans, we also call it Creutzfeldt-Jakob disease. So this slide is based on page 192. It will be asked for. So prions, it's not living, it's not a virus, it's a protein. So prions as a protein causes mad cow disease or Creutzfeldt-Jakob, and basically turns your brain into mush. Okay, now draw a line. The last slide before I clarify everything is emerging diseases. I'm going to change the title to this guy, emerging diseases. That is what we're seeing. So in 2020, when we came across the coronavirus, or in 2002, we came across SARS, or near around 2005 or so, we came across the MERS in the Middle East. All of these are because of emerging diseases. So there are new diseases coming up because of what is happening to the environment. So emerging diseases. Next slide. Don't care. Don't care. Don't care. Don't care. Don't care anymore. This is a nice comparison to keep in mind between DNA and RNA. It's mentioned on page 193 at the end of the chapter. Okay, next slide. This is one I put up before regarding semi-conservative. So that's something we mentioned before. Okay, don't care. Here's another picture, but we've already seen it. This one I don't care for. This one I don't care for. Now, before I end this video, make sure you watch within the PowerPoint. First, you watch the video on DNA replication. Make sure that's clear. And then once you have DNA replication down, then you can move over to transcription and translation as a different story. If you have any questions, please ask me as part of our conference.